Hi everyone, and thank you for joining us today. We're here to talk about critical design considerations for rapid diagnostic tests in a COVID-19 pandemic. So we're joined by a group of panel guests. So in no particular order, we've got Brendan O'Farrell, the president and co-founder of DCN Diagnostics. We got Darren Rowles, who's the president and CEO of Sona Nanotech. Klaus Hochleitner, who's our technical specialist at Cytiva, and David Wilson, who's the commercial director at Avacta. The format of today's call is we're going to have a discussion around 3 points. So understanding the different technologies available, moving on to why a dual approach between those different technologies is the best approach, and then spend some time around the different design challenges that people will face when developing lateral flow tests and what support exists to help developers. So to start, Brendan, would you like to help explain the different technologies available?
Sure thing. Thanks, Lee. So I guess the methods for testing relatives to SARS-CoV-2 infection really break down broadly, I suppose, into molecular versus immunoassay. So molecular tests, we're looking for obviously the molecular material of the virus. In the case of SARS-CoV-2, that's RNA. So the primary method used is RT-PCR, and that starts by converting the viral RNA into DNA and then amplifying it using PCR to a level at which it can be detected. So several other isothermal amplification methods obviously are being developed and implemented as well. So the main advantage of RT-PCR in this context is the relative speed of the assay and the fact that it can be automated and high throughput. And it can be very sensitive and specific if it's developed correctly. So, but obviously RT-PCR has its limitations as well. The primary sample used for detecting SARS-CoV-2 is a nasopharyngeal swab. And those are difficult to collect effectively, and they're really uncomfortable for the patient. Taking them also potentially exposes the healthcare worker taking the sample to infection, which has pushed us in the direction of a lot of thoughts around self-sampling. Other sample types like nasal swabs and tongue swabs are being evaluated and implemented, but they obviously come with their own challenges as well, particularly when it comes to the performance of the assay. So traditional RT-PCR requires a lot of lab infrastructure, a lot of equipment, trained users, and these tests are most commonly done at scale in central or hospital labs as a result. So more recently, we've seen some small instruments developed that can be deployed to more decentralized testing environments. And these have the advantage of a lot more portability. But obviously, they're complex instruments, for the most part, still requiring trained operators and at least some infrastructure and maintenance. So there's also the primary issue, I suppose, of access, time to delivery of results and scale. And turnaround time right now on centralized RT-PCR assays from sample to result is on the order of multiple days, even in the best of circumstances. So processing of COVID-19 samples also requires biocontainment labs operated by highly trained technicians, obviously. And for more resource-limited countries with less access to central laboratory resources and less capacity, than the developed nations, this kind of central lab testing at the required scale really isn't an option. So the alternative approach is immunoassay, and with this format, we're detecting the presence of specific proteins in a sample that are indicative of current or previous infection. And these assays can either be instrument-based and run in a central lab, or they can be run at the point of care as a rapid assay. The most ubiquitous format of rapid assay being the lateral flow format, of course, that most people are familiar with in the context of pregnancy testing or HIV testing or strep throat testing at the doctor's office, but has been applied as a very mature technology in literally hundreds of applications over the course of the last 30 years or so. So there are two primary applications of the immunoassay in the context of the pandemic. I suppose there's antigen detection and antibody detection. There's a lot of different use cases around each type, but in broad strokes, the antigen detection indicates the presence of viral proteins in the sample. Once again, typically the nasopharyngeal swab in the current embodiment. And the presence of these protein antigens, like the spike protein, is indicative of infection with the virus. So the value of the antigen testing is probably self-evident. If a swab taken from a patient tests positive, they're infected and likely infectious. On the serological assay or the antibody detection assays, these use binding reagents to detect the presence of antibodies directed against the specific viral antigens. So typically we're looking at IgG and IgM, although there's growing evidence that IgA has utility in the context of COVID. So serological assays are useful because a patient that's recovered from COVID-19, once the virus is cleared from the body, the viral RNA is no longer available for detection in the respiratory tract. So there's really a pretty short window during the acute stage of infection in which SARS-CoV-2 can be detected using PCR or immunoassay for viral antigen detection. So while the PCR or antigen detection by immunoassay works well for diagnosis of ongoing infection, it doesn't give a clear indication of whether a patient has been infected historically and potentially, and I would put a lot of emphasis on the word potentially, what their immune status might be. I'm sure we'll come back to that. But antibody tests have their limitations too. The data is still emerging. It appears that the antibody response to COVID-19 is slow compared to a lot of other infections. And with limited data, it appears that the initial IgM antibody response doesn't peak until about 9 days after infection, and the IgG response is several days after that. And to put this into perspective, most viruses would elicit a primary immune response within about 5 days of infection. Now, because of this, SARS-CoV-2 antibodies are unlikely to make good markers of acute COVID-19 infection. So, as a result, the detection of antibody alone for acute phase diagnosis isn't really viable, and the topic of whether a total antibody test is the ideal specification rather than one that differentiates IgG and IgM is being pretty hotly debated in places. But while antibodies, might not be appropriate for acute phase diagnosis, they still show a lot of valuable applications in the context of COVID-19. So potentially the most valuable use of wide-scale antibody testing is as a public health tool, indicating levels of exposure to the virus. There's lots of talk about using these assays to indicate immune status, but the science is still largely out on whether that's a valuable use case for a lot of these tests. But to date, I'm aware of at least 12 different identified use cases for serology assays, a lot of which require somewhat different assay specifications. So how to use these assays and develop them is going to be a hot topic for some time to come. So hopefully that outlines the basic framework. There's lots of details and nuance in there to be debated and discussed, but I'll hand back at that.
Yeah, no, thanks, Brendan. That's brilliant. So I guess, from that explanation, Klaus, are you able to help explain how, I guess, we're using both of the molecular and immunoassay approach in the real world to tackle this?
Well, the point with the molecular diagnostic test was that these were the first tests that had been developed because people quickly sequenced the virus, created primers, and were able to detect the virus in patient samples. As Brendan already pointed out, that's something that needs to be done in a centralized lab. It's something where you need an infrastructure, train people, you have to ship the samples to the lab, and it takes ages before you get the result back to the patient. And that means in that time, you either have to quarantine the patient or the patient will spread the virus to other people. The development of tests for antigens, for viral antigens, is ongoing. So first test on the market was limited sensitivity, but the original idea is to have a test that identifies the patient as positive, let's say in a doctor's office, and is then confirmed by a molecular diagnostic test in a central laboratory. So that would be a two-step approach. And that should avoid at least false positives. The risk is false negatives. And the tests that are currently on the market, the best one has a sensitivity of 80%. That means of 10 people that are real positives that you test, you will miss 2. And that means in a moment as a standalone test, it's not yet feasible. And this is what people are working on. What makes your life an additional misery is that there are 4 endemic human coronaviruses that cause 10 to 15% of human common colds. These viruses are all closely related, so you have a fair chance of getting positive signals that are, from a biologist's point of view, real positive signals. And unfortunately, you detect the wrong virus. Things will get worse when we talk about antibody tests. The first problem is that in a typical study, what you see is that the first IgMs appear on day 5 after onset of symptoms. And then you have 40 to 50% of the patients developing the antibodies. You will have IgMs in all patients around day 9 or 10, which makes that, as Brandon already said, a no-go for finding acute phase diagnostics. And what is even more interesting is that antibodies against different coronaviruses cross-react. This has been published for SARS-CoV-1 and antibodies against the endemic human coronaviruses. We know it for the MERS coronaviruses. It's the same story, and it goes in both directions. And there is absolutely no reason to believe that this will be by any means different for SARS-CoV-2. And that means if you go for antibody tests, you have a very high chance to get an awful lot of false positives. That means you will run into trouble with your epidemiological statistics. At the end, it means we need to develop more accurate tests for the antigen in order to help diagnose COVID-19 in under-resourced regions and even in what we call first world countries, in a near patient situation in a doctor's office, and then confirm that by a PCR test. What I do not buy in, to be honest, is the idea to have self-sampling. Self-sampling, regardless whether you use nasopharyngeal samples or saliva samples, has a very good chance to fail due to user errors, and that makes that very difficult as a screening test, and this is what we are looking for. And that means I would always opt for a test that where the sampling is being done by a trained person, which leads us back to the doctor's office. So that's for the approach that is currently being used. So do an immunological antigen test first, and then do a confirmatory test for the positive tested people. We have to develop antigen tests that have a sensitivity that's much better than 95%, else it will not be useful for a screening test. And the antibody test that we are talking about will not involve a two-step approach, but they have a fair chance to give you false positives due to cross-reactivities with the endemic human coronavirus antibodies. Again, this needs to have a very high sensitivity and specificity in order to be useful as a screening tool to talk about immunity in the population, whatever that means. And nobody knows how long it will last. The problem is we know from all other coronaviruses it doesn't last forever. And again, there is no reason to believe that this will be the case, that this will not be the case for this new coronavirus.
Okay,
end,
thank you, Klaus.
Yeah, we will need to test for a very long period of time.
Yeah. Okay. No, thank you, Klaus. I think that you said quite a good picture of how complicated this is to get something with the required level of sensitivity and accuracy that we need, which I know you touched on a few different areas there, you know, such as sample collection and other pieces, but maybe David, you can help start the conversation around some of the design considerations and some of the challenges that typically you've encountered during development.
Yes, so at Avacta, we've been focused on the development of a antigen test on a lateral flow platform, and recently announced our collaboration with Cytiva to develop that particular test. The aim is to develop a test that can be easily deployed in a community, primary care, and a consumer setting. We believe the lateral flow format is really the only format that we're aware of that will enable the mass testing or mass population screening that's going to be required in order to get society back to normal, whatever normal will be. And there is a lot of demand for this type of testing outside the traditional healthcare settings. One of the biggest opportunities is to enable workforce testing, for example, to get workers back into companies, to get sporting events back, concerts, public transport. So there's a huge demand. So we've been focused on the development of a test which will enable that to happen. So that requires what we think is a lateral flow format and saliva as a sample, as opposed to the nasopharyngeal swabs. So some of the challenges, well, one of them is time. Typically these tests take many months, 18 to 24 months to develop. We don't have the luxury of that at the moment. So all companies I know that are developing these and other COVID-19 tests are trying to do it in a way that's different from how it's normally done in order to accelerate the time to market. And clearly that introduces a somewhat risk. But we don't have the luxury of 18 to 24 months to get these products out. So that's one very big challenge. Also, the access to clinical samples. They are in high demand and short supply, increasingly so as the pandemic starts to disappear. Although I agree with Klaus's earlier comments that the need for testing and the demand for testing is not going to go away for a very long time. We anticipate that there will be a need for testing, in very significant numbers for at least another 12 months. So time is a challenge, and develop a test that can be deployed in these settings is also one of the biggest challenges for a diagnostic test developer, because of the need to develop a test that can be used easily by a consumer, for example, for a self-test. So that's a challenge. It's also a challenge from the regulatory approval process. Typically the regulatory approval for these tests and the clinical validation study needs are significant. So again, the authorities, the FDA, for example, the MHRA in the UK, are looking at ways to provide accelerated approval processes for these tests so we can enable them to get to market. Otherwise, again, they won't be coming to market in time to enable us to fight this pandemic. So there are a lot of challenges associated with the fact that we're trying to get something to market much more quickly than we would traditionally do as a diagnostic product manufacturing company, and to be honest, are probably more comfortable doing with the more standard timelines. But these tools are absolutely required. Without them, it's going to be much more difficult for us to get society back to normal. So there's and then on top of all of this, there's supply chain issues. I mean, I'm outlining lots of problems, but they're really just to introduce maybe topics of conversation for the rest of the webinar. Because of the demand for these tests, the supply is under a lot of strain, and that's the supply not just of the tests themselves, but all the components and raw materials. And that's an issue which needs to be addressed because the supply chain needs to be ramped up again in a time scale which it's never been done before. So yeah, it's just an outline of some of the major hurdles and challenges that we're facing at the moment to get these products to market.
Okay, thanks, David. Darren, is there anything you'd like to elaborate upon what David's added, or anything you'd introduce from your side?
Well, yeah, thanks, Lee. Well, as David outlined, I think everybody listening to this can probably get a sense of the enormous challenge that anybody that's developing one of these tests is up against, for sure. And it does come down to time constraints. That's a major thing. David outlined, all the problems that we're seeing. So I'm not going to kind of dwell on those. They'll be discussed as we go through this presentation. I think one of the major things that SONA has come up against with regards to challenging, in terms of our biggest hurdle, is probably that access to some of those materials. Be it everything from test component materials all the way through to some of the specific biological materials that you need to get to be utilized. Similar to the Avacta test, SONA's test is being based around an antigen-based test. Initially we're focusing on the nasopharyngeal swab type systems. But our platform and our process is easily adaptable to other matrices, including saliva as well. But our first pass, our first priority really is to kind of see a critical path to developing a test utilizing a nasopharyngeal swab. So alongside all the other kind of constraints that David kind of outlined around time, regulatory, clinical pathway, access to materials, etc. I think we echo one another, and we've spoken offline around these things many times. One of the other major things, probably that is going to be a challenge, is we're certainly a topic of discussion that David just touched on, is the infrastructure around the development of rapid tests. Now, typically, lateral flow tests, hundreds and hundreds of millions of these tests, produced every year across a plethora of different disease states and for many other use case scenarios. But there's probably maybe a handful or two of core material suppliers for things like membranes and pads and antibodies even, but then also machinery to produce these tests. Unless you have a significant production setup in-house, which obviously some of the major players do, but everybody else doesn't, and they utilize CMOs that are out there, contract manufacturing organizations, and again, there's typically a handful of those that obviously we're in discussions with multiple groups around that, and one of those challenges is around beyond the test development process itself. You then typically would go through a tech transfer process, which again, normally we talk about normal versus expedited timeframes. That process is normally a 6-month process in itself, and we're asking those CMOs to pretty much rip up the rule book, but still make sure that they're adhering to those quality systems to make sure products that are transferred are done so correctly and then produced at the scale of manufacture that is expected, because as we know, the mass screening that's required here, that's expected to ramp up production from even from a few million tests to hundreds of millions of tests, which is what we all anticipate is needed. It's a huge task on not just us as test developers, but also on the manufacturing organizations as well. And those manufacturing organizations are typically serviced by, again, a handful of companies that produce the manufactured equipment. So it's as a diagnostics industry as a whole, everybody that's in this tight network have all recognized the challenges. We've all kind of everybody's come together very, very quickly. What were once seen as competitors are now certainly collaborators. Everybody's working hard together to kind of allow each other to bring those tests to market, because everybody can see the need for that.
So supposedly, just to pick up a little on the topic of development challenges, the DCN Diagnostics is a contract developer of lateral flow assays. This is what we do for a living. It's what we've done for several decades now, and so we're used to dealing with the typical challenges, right? That everybody in the industry is aware of. Starting from the concepts of designing really usable tests for point of care application, which covers everything from getting the right sample to getting the test to run correctly, to making sure it can be interpreted correctly digitally or by eye. to the analytical challenges of just making the system work, selecting the right reagents, getting the right materials, ensure that everything, as Darren and David said, goes through the correct process and goes through it correctly, ultimately ending up in a properly validated product that can then be scaled and manufactured, right? So all of those challenges still exist here, but as I say, obviously we're trying to compress this timeline. And we're doing it based on a real risk-benefit analysis, right? So there is risk to this kind of acceleration. And the reason we go through that usual process of development is to end up with a really robust, reproducible product that can be scaled, right? And as Darren pointed out, that scaling and that manufacturing requires a very, very specific and quite limited skill set. So one thing that in this whole process that can't be clawed back, obviously development happens on a continuum. So we can draw the lines anywhere in the process to say, we've reached a point where we're happy to release this product to market. We've reached a point where we're happy to submit this to a regulatory authority under an emergency use authorization or whatever the case may be. But what we can't claw back in that process is time, right? So we are drawing a line in that development process at a point where we haven't, by definition, under any circumstances, demonstrated that our products are highly reproducible and highly scalable at the level at which we're going to have to manufacture them. So there's inherent risk in that process. So what we have to do as development organizations and manufacturing organizations is try to pull back as much of that risk as we can through, still the proper application of development methodologies, and understanding that even after we have released these products to market in this specific instance, development continues. Right. There's not a defined line here that says this product is done. It's the exact opposite, in fact. From every level of this process, the fact that the target product profiles are still shifting, both for antigen and for antibodies. So we actually don't really have a great idea of what the final product has to be. We're making some very well-educated guesses. But that's going to change. So the target product profile is going to change. The performance requirements are going to change. Our understanding of the process is going to change. Our experience with these processes is going to change over time. So this is going to be very much an evolutionary process. And one of the things when we're talking about supply chain that we see as, I guess, one of the bigger risks, certainly we all understand the reagent supply issues. We understand the material supply issues. We understand the manufacturing equipment side of things. One of the biggest gaps we see in the market is knowledge and its education. A lot of companies have been forced into a position where they're having to scale manufacturing processes really quickly. They're having to do so with people who may not have the experience level that we'd like to see, in these processes. And again, there's a risk benefit being done here. We need these products. But I certainly wouldn't skip over the question of the knowledge gap that's in this market right now. We need more trained people. We need more experienced people. And that's unfortunately not something that's going to happen quickly, but it's something that we need to work on too.
No, I think it's very good. And one of the things I was going to add, I know that you touched on it very briefly earlier, was about the importance of time to market, the time to test, and getting a quick result on these, which, it's critical that we have a instant result where the current bottleneck is taking several days sometimes for results. Do you want to help expand on that piece that you mentioned earlier, Brendan?
Sorry, Lee, you actually broke up on me there just a little bit. So the question was on time to results?
Yeah, when we're talking about the different technologies and you introduced that, you're saying that lateral flow, the importance of this is, getting a quick result. And I guess, there's some of the challenges around that, I guess, in what you're saying about the less reproducibility and that you're able to go for at the moment.
Well, the time to results, obviously, in the context of the testing that we're doing, time to results is critical, right? So one of the benefits of lateral flow technology, as a platform, is that allows us to take a sample, do the test on the spot, and have a result within minutes. Now, we can couple that, obviously, with reading technologies as well, whether they're, within the cassette, whether they're external readers, and do instantaneous data gathering, databasing. We can have all of that data available in real time as well. So there's obvious benefits to that in the context, particularly of antigen testing, where we're trying to get to a decision as to whether somebody has an active infection really quickly, get them into quarantine, take whatever measures need to be taken to ensure their health and safety and that of the people around them. And so having that instantaneous or virtually instantaneous result is a really, really huge benefit. Now, in the context of, mass population screening, particularly in the US, the US has growing capacity for centralized laboratory testing, where we can take those samples. Turnaround times will improve, but we're still talking about probably days from sample collection to delivery of that result. So in the context of antigen testing, that still introduces a gap where there's significant risk. Now, in the context of antibody testing, where we have so many potential use cases that people are looking at, and some of them do require that instantaneous result, right? So some of the back to work testing, or testing of healthcare providers, or whatever the case may be, we need that result, right? And this, both from a serology and an antigen perspective, I think the lateral flow platform provides probably the only available solution. So it's really ideally suited to this in many ways.
Okay, thank you, Bernard.
Technology, that's correct. My problem is that we need specificities and sensitivities on an extremely large scale that is definitely unprecedented, simply due to the fact that's a massive screening test, and this has never been done before. In all lateral flow tests that we have on the market, you have patients that show symptoms and that you want to test. What we want to do now is we would like to test people that have no symptoms. We have not the slightest idea whether they have or had an infection, and we need a test that gives us an accurate result on answers to these questions. And that's a completely different beast. And the second problem is this virus is a new virus, and that means it's a rapidly moving target. Currently, I'm seeing about 50 papers at least per week published on preprint service, so you can't read them all. But the knowledge that is gathered and the surprises that are appearing as really astounding. And we as test developers or material developers or reagent developers have to try to take pace with it, and that's a real challenge.
And from an algorithmic perspective, one of the biggest differences here is that we're talking about utilizing a serology test in a way that it's not normally used, right? So serology tests are usually reflexed to some kind of an orthogonal message or method for confirmation, right? Depending on the endemicity of the environment they're used in. So in this case, we may not have that safety net. So it's different.
Yeah, it's different. There's absolutely no safety net.
Yeah, I think the sensitivity issue, Klaus, that you brought up, and we've had numerous conversations about this over the past few weeks. As you say, I think it's a major challenge, for anybody developing a test, trying to get that sensitivity level down. I think we've discussed whether you need to get viral load readings down to the 10 to the 2, 10 to the 3 level, just to get a sense of the limit of detection of these tests, and if not lower, to detect viral loads in patients, because say that differentiation between symptomatic or pre-symptomatic patients and asymptomatic patients is a mass difference, when it comes to screening, because I think, even pre-symptomatic patients, it's hard for them to get a test in many countries, that certainly don't have huge capacity for centralized lab testing. So I think screening tests like the ones that we're all trying to develop here, whether it be antigen or antibody based, certainly have their role to play in it. And as we've alluded to before, that risk benefit, kind of trade-off is the main key thing. But again, coming back to some of those technical challenges, the sensitivity is going to be the major issue, and there is that specificity issue around that cross-reactivity interference, with the existing kind of SARS-based viruses that are floating around in the environment. So again, beyond supply chain and organizational issues, there's actual technical issues that simply need to be overcome by everybody as well. and they pose their own challenges. But I think the world, and certainly our space, is full of smart scientists, and we're all working hard to overcome those issues. And I think we're, I'm sure together we'll all be able to do so.
Yeah, that's right. I think specificity is something that many people overlook at that point. Let's assume you have 5% of the population with an active infection that may or may not show symptoms. So that's the big problem. And you have a specificity of 95%, which means that 100 negative patients, 5% will come up with a false positive. The outcome of that is, even if you have 100% sensitivity, that it's a little bit more complicated mathematically, but what you would have is that from all of these positive tested people, 50% are false positives. And that's a problem, because everybody would say, oh, come on, what is this for a test? Because when you run the confirmatories on a PCR test, for example, 50% of these patients turn out to be negative, but you have quarantined them. You have probably looked for contact persons. You have put a quarantine on the contact persons, and you panicked an awful lot of people. And at the end, people will say that the test, the rapid test, is nuts. And that's also something that needs to be considered.
Yeah, and just building on that, Klaus, obviously when you're looking at trying to get, the best test will be high sensitivity, high specificity, but often it's a trade-off, isn't it? When you're trying to get, to get high sensitivity, often you have to give something on specificity and vice versa. I think for the antigen test, the bias will be, obviously sensitivity and negative predictive value. It's not ideal having too many false positives, but I think it's even worse having too many false negatives. At least with the false positives, there may be a way to introduce a confirmatory test for those, but the false negatives, you would never know. To have someone walking around thinking that they're not infected when they are, would be a much more difficult problem. So I think if you have to go one way or the other, then for the antigen test, the bias should probably be towards sensitivity. Yeah, ideally, yes. High on both levels, but
yeah, definitely yes. So in an infectious disease test, regardless what we are talking about, it doesn't necessarily have to be SARS-CoV-2 and COVID-19. In an infectious disease test, what you're looking for is the best possible sensitivity, because you do not want to see any false negatives. You will never get 100% sensitivity, since there will always be clinical outliers that you simply will never find, regardless how good you do your clinical validation. But the difference we have here is in just opposite to any other infectious disease test that is on the market, is we want to do a screening test, and that makes specificity a bigger hurdle than usual. Let's assume you look for you do a hepatitis B test, and you have reasons to do the hepatitis B test. And if you have a 95% specificity, the few false positives that you have the number will be much lower than the real positives that you have. In the screening test, a 95% specificity can easily bring you to more false positives than right positives, and that will give you problems. Not necessarily on the market, but you will run a large number of confirmatory tests that come back negative, and as said, you will create a lot of trouble with the patients, with their environment, their personal environment, their work environment. So you may shut down complete factories due to false positive tests, and that's the worst case, and that's something we need to deal with.
Yeah.
Okay. We've got about 5 minutes left, and I think that I need to jump in, because you could probably spend another half hour or so, probably longer. It's still going round with the team on the call, so maybe we can cover some more in the questions that come up from the audience. But maybe as a way of closing the call and summarising, if we can go around each person maybe in alphabetical order, So starting with Brendan, this may be any advice you could give to people looking to develop these tests, or what support is available that you could help advise them through to?
Well, I mean, there's an enormous amount of support available, so particularly, the US system is the one we know best. There's a lot of support in the US system right now, coming from NIH, coming from BARDA, coming from a variety of different areas, both from a funding, from a networking, and from a technical perspective. Within the industry itself, there's a network of core providers that have historically existed within the lateral flow space. Companies like ourselves that are on this call, and I think we have historically all had a culture of both promoting the best possible practices within the lateral flow industry and assisting companies coming into the industry. So there is quite a bit of technical resource available from the materials providers, from the reagent providers, from companies like ourselves that are contract developers and educators and trainers and contract manufacturers. So there is quite a bit of support already out there inherent in the industry. Everybody is strapped from a bandwidth perspective. Everybody is busy right now. But at the same time, we all recognize the importance of this. And I think as an industry, one of the most heartening parts of this, from my own perspective, has been watching the reaction to this from the service and materials and equipment side of the business, from the people who really facilitate the development and manufacture of these. The industry has really come together in quite an incredible way. So I think anybody who touches really any one of the core suppliers in the industry is going to get networked very quickly into the other aspects of what they need. So, we're here to support both the companies that are newer to the industry, as well as the more mature developers and manufacturers, or that are just under pressure with this whole thing to have to develop products while validating them, while standing up new manufacturing lines and new facilities, while training new people. We're all here to help as best we can.
Okay, thank you. And Darren?
Just to, I guess, in Brendan's comments really around how that network has all come together, it's really encouraging. It's really heartwarming. I've been part of it for nigh on 15 years in various guises, through contract manufacturing organizations to now kind of heading up a test development group ourselves. And I found that network that I kind of got in touch with over the years and built, has certainly kind of come to fruition in helping us deliver this test to market as quickly as we've been able to. My advice to anybody kind of stepping into this is, initially look at what's already in the market, to determine whether, is there an opportunity here for me to compete in that market with that test, albeit that, we're going to be testing for COVID, SARS-CoV-2 for a long, long time. You want to make sure that you're in a position, to make sure your business is competing there. So once you've identified which area you want to kind of work into, get in contact with as many people as you can within this network, be it somebody like Brendan at DCN to help with that test development, be it with people like Klaus, and yourself, Lee, around those membrane suppliers, and we'll then certainly put you in touch with all the other key suppliers for materials. Reach out to people like myself and David, who supply some of the reagents into the network as well. Because you're going to need that. You're going to need that support. As well as the end manufacturers, and even regulatory and quality support as well. One of the first things we did when we started doing this was reach out to the FDA and get that contact in there, understand what they needed as part of this and what they were going to do. And they held their hands up and they were like, we don't really know yet. Tell you what, let's work together and try and figure this out, which was great in one way. And it helped us kind of steer our path down that critical path. So certainly, use the network that's there. It's a fantastic and heartwarming network that's accessible to everybody, and they'll embrace everybody with open arms.
Oh, thank you, Darren. It's really insightful. David, anything to add from your side?
Well, I mean, I think Darren and Brendan have pretty much covered it, but I would just like to add that that sort of network and collaborative effort extends to the different players, different companies in the industry itself. I've been in the industry for many years, as we all have, and I've never experienced such a collaborative effort. And putting aside some of the normal competitive issues, there's a collective effort to get these products to market as quickly as we can. So that's heartening. I also think by going through this now, the industry is going to be more pandemic ready next time, because there will be a next time, and there may be a next time for this current pandemic, but there will be another pandemic. And I think what we've gone through in terms of getting ourselves prepared to develop products which will help tackle this pandemic will put us in a very good place for the next time it happens. We'll be able to respond much more quickly. And I think for the industry as a whole, this has been it's an opportunity for the industry to showcase what we can do and what value our products actually have, because that, I think, has been somewhat under the radar for many, many years. And so I think those are many of the positives. So someone coming into this, I think, would find an awful lot of support from all the different sectors that Brendan and Darren have said, including companies that they would typically see as their competitors.
Okay, excellent. Thank you, David. Klaus, anything briefly from your side, because we're about to reach the Q&A.
So some famous last words. Yeah, I could bore you with another comment on networking, and I will definitely do that. So what all of us have told you is, yes, networking is key here. We talk about an unprecedented situation, about a pandemic that will, and nobody will able to deal with that alone. So the advice is, get out of your ivory tower. Don't try to reinvent the wheel on your own. And talk to people, talk to reagent suppliers, to material suppliers, and work in a network. And that's the only chance we have to get this pandemic under control, to have tests developed that really do the job that we want them to do, and to be prepared for another pandemic or the reappearance of SARS-CoV-2, because if there's one thing that is sure, it's not over yet, and this virus will stay with us. So there's enough work to do.
Okay, excellent. And I guess that brings us up to our Q&A deadline. So thank you, everybody else. I think it's been excellent. Really appreciate your insights and your input, and I'm sure the audience and everyone joining this session will echo that comment as well. So thank you very much, and I think we'll end here and hand over to our Q&A.